Welcome to this explainer. Okay, so today we are tackling one of the most notoriously feared biochemical pathways in your entire physiology syllabus. If you've ever stared at a textbook diagram of the Ornithine Cycle, sometimes it's called the Urea Cycle or the Krebs-Henseleit Cycle, and just felt your head start to completely spin, well, you are in exactly the right place. Today we're going to take this giant puzzle and make it incredibly simple. By the time we're done here, you'll literally be able to map out this entire cycle with your eyes closed. We're grounding our explainer today in its original educational context, as our source material beautifully puts it in Hindi, which translates to, I hope everyone is studying with all their heart. We know exams are right around the corner. And hey, when the pressure's on, these long biochemical cycles with all their crazy names can feel totally overwhelming. But don't sweat it. We're going to break this down logically and visually so it sticks in your brain for good. Go ahead and grab a pen and paper right now. So let's dive in. Why does this complex cycle even happen in our bodies every single day? Well, as we break down proteins and amino acids for energy, our body produces a byproduct called ammonia. And here's the absolute kicker. Ammonia is highly, highly toxic to our cells. Basically, our own body is constantly generating a poison that it desperately has to get rid of. To figure out how we survive this, here is our roadmap for today. First, the toxic ammonia problem. Second, we'll go inside the mitochondria. Third, out into the cytoplasm. Fourth, the magic of urea. And finally, number 5, your exam magic key. Let's start with section 1, the toxic ammonia problem. Now, think about the animal kingdom for a second. Aquatic animals, they have a huge evolutionary advantage here. Because they literally live in water, they can just continuously flush that toxic ammonia right out of their systems. They're what scientists call ammonotelic. But us land-dwelling terrestrial animals, we absolutely cannot afford to lose that much precious water just to flush out our waste. So instead, we have to rely on our liver to perform this amazing rescue mission, converting that toxic ammonia into a much safer, super water-soluble form called urea. Moving on to section 2, inside the mitochondria. Before we get completely lost in the chemical weeds, I want you to see the big picture. Here's a high-level outline of all 5 steps of the cycle. We're basically going to track our toxic little molecule as it morphs and changes through these 5 exact stages. All right, step 1. We are starting deep inside the mitochondria of a liver cell. Here, our villain, the toxic ammonia, is smashed together with carbon dioxide gas. This fusion creates a brand new compound called Carbamoyl phosphate. But here's the thing, combining these two is a serious uphill chemical battle. It doesn't just happen magically on its own. And what's super fascinating here is the biological toll it takes. Because it's an uphill reaction, the cell has to actually consume its own ATP energy just to force this reaction forward. Think of it like paying a toll at a biological toll booth. This isn't free. Your body is literally spending its hard-earned energy just to keep you safe from that ammonia. Which brings us to step 2. Still hanging out inside the mitochondria, our newly minted carbamoyl phosphate reacts with an amino acid called ornithine. Boom, they combine to produce a new molecule called citrulline. And honestly, this right here is exactly why this whole pathway is famously called the Ornithine Cycle. As you're going to see at the very end, ornithine is both the starting point that kicks things off and the final product that gets totally regenerated. It's the ultimate biological recycling program. Okay, section 3. Out into the cytoplasm. Right, so we've officially crossed the cellular border into the cytoplasm for step 3. Once we're out here, that citrulline molecule bumps into another amino acid called aspartate. The two of them lock together to form a massive new molecule called argininosuccinate. But guess what? Time to pay up again. This bonding process out in the cytoplasm isn't free either. It consumes yet another molecule of ATP. So your liver's actively spending energy at multiple different stages just to build this giant argininosuccinate molecule. And now for step 4. Because that molecule we just spent so much energy building is frankly too huge and unwieldy, a specific enzyme swoops in and chops it right in half. It splits it into 2 completely separate parts, arginine and fumarate. Now, the fumarate actually waves goodbye and leaves the cycle entirely to go join the famous Krebs citric acid cycle. But the arginine, it stays right here for our grand finale. Let's get into section 4, the magic of urea. Here it is, step 5. Water gets introduced to our remaining arginine molecule. This sets off a hydrolysis dynamic, which is just a fancy way of saying we're using water to trigger a chemical split.
This reaction brilliantly snaps off the finished, perfectly safe, highly water-soluble urea molecule. This is it. This is the exact non-toxic waste product our terrestrial bodies can just effortlessly flush away without wasting all our water.
But wait, what happens to the leftover piece of the arginine after the urea gets popped off? Well, it leaves behind our old friend, the original ornithine molecule. This regenerated ornithine then travels straight back across the border into the mitochondria, totally ready to grab the next incoming carbamoyl phosphate. The entire 5-step cycle just loops and starts all over again. Finally, Section 5, your exam magic key. Okay, listen up, because this is absolutely crucial for your test prep. The worst thing you can do during a high-pressure exam is waste time writing pages of dense paragraphs. Trust me, don't do it. Instead, memorize this checklist to draw a perfectly clean flowchart. First, literally draw a line on your paper to represent the mitochondria boundary. Show steps 1 and 2 inside and 3, 4, and 5 outside in the cytoplasm. Next, write the enzyme names right there on the arrows. Make sure you clearly label the 2 specific spots where your body pays that ATP energy toll. And then explicitly show step 5 where the water enters and the urea drops out. If you give the examiner a diagram like that, they're going to hand over full marks without reading a single word of text. Before we completely wrap up this explainer, let's do a really quick pop quiz to see if this is sticking. Drop your answer in the comments below. In which organ of the human body does the ornithine cycle primarily take place? If you've been following along with our little biological tour, you should know this one instantly. To close things out today, our source material leaves us with a really wonderful reminder for everyone studying hard right now. Stay disciplined during these final days of test prep for sure, but please do not forget to take care of yourself. Your brain simply cannot memorize these intense biochemical pathways if you aren't fueling it with decent food and letting it recharge with enough sleep. Think about it this way, and this is the final thought I'll leave you with. If your liver is literally working overtime every single day, spending its precious ATP energy just to keep your internal environment clean and safe, doesn't it make sense that you're obligated to treat the rest of your body with that exact same level of care and respect? Keep learning, take care of yourselves, and I will catch you in the next explainer.